Ladies and gentlemen, as we are about to begin, I introduce myself first. My name is Nurul Nadia, and it is my pleasure to be your MC this morning. Assalamualaikum and good morning, everyone. Welcome to the Agro-Food Productive Webinar on important infectious poultry disease in Malaysia. We have here Dr. Yap Teow Chong as our moderator today. He is a veterinarian graduating from the University of Melbourne in 1972. His postgraduate studies include a diploma on food hygiene and veterinary public health from the Royal Agricultural University, University Copenhagen, and a master degree from Burdock University, Western Australia. He has worked in the Department of Veterinary Services for 14 years, 20 years in the Malaysian poultry industry, and 10 years with WHO and FAO as an international expert on AI and food safety risk mitigation in poultry supply chains in various countries. Currently, he is now a technical consultant to FLFAM. Now, without further ado, I will now pass the session to Yang Professor Dr. Please welcome Dr. Yap.
Thank you, Puan Nurul Nadia, our wonderful MC for the day. And welcome participants to this morning's webinar on important infectious poultry diseases in Malaysia. And we hold this webinar series all about poultry industry, starting from transformation of the industry to food safety, as well as to public health concerns. And today our topic, of course, will be focused on important poultry diseases. Now, poultry diseases are not new to the country. Some farmers have known them for many years, decades, and have already taken very good precautionary measures like vaccination, particularly like Newcastle disease. But however, there are some diseases that may not cause outbreaks or high mortality, but they are nevertheless very important in this modern age of poultry production. Because in our poultry production today, we have to be very careful with our flock health management so that we stay on top of our productivity. So we must have a good prevention and control on all infectious diseases. Today, we are very fortunate to have two very distinguished speakers to talk to us about three important poultry diseases that somehow may still not be fully covered. Now, our first speaker would be Professor Mohd Hair Bejo. Prof. Hair is a graduate in veterinary medicine from University Putra, Malaysia, and he has focused his attention in the field of veterinary pathology with a PhD from the University of Liverpool in England in 1990. He had also been a previous dean in the Faculty of Veterinary Medicine, as well as he's a director of the Putra Science Park in UPM. He's currently also the Professor of Veterinary and Avian Pathology in the Faculty of Veterinary Medicine. Prof. Hair, besides his career, he's also very active in being the president of the World Poultry Science Association, or WPSA. He's founding fellow of the Malaysian College of Veterinary Specialists, and also president of the Association of Institution of Tropical Veterinary Medicine. So we are very happy today that he'll be talking to us on two subjects, and it is foul adenovirus infection or inclusion body hepatitis, as well as later another topic, infectious bursal disease, or commonly known as Gumboro disease. I would like Dato to do his presentation. Over to you, Dato.
Bismillahirrahmanirrahim. Thank you very much, Dr. Yap, for your very kind introduction. Assalamualaikum warahmatullahi wabarakatuh, and very good morning to all participants, ladies and gentlemen. First of all, I'd like to thank the organizing committee for inviting me in this seminar. It is my pleasure to share with all of you on the issue, challenges, and strategy for the prevention and control of fall adenovirus infection in chicken. As many is aware, the family of adenoviridae in poultry is classified into Group 1, Group 2, and Group 3. And Group 1 fall adenovirus is a causative agent of inclusion body hepatitis, hydroperoxidase syndrome, gizzard erosion, which affect broilers, layers, and breeder chicken. This virus is a double-stranded DNA virus with non-enveloped structure comprised of 3 major structural proteins known as hexon, penton, and fiber involved in the replication of the virus in host cell. As a non-enveloped virus, this virus is highly resistant to physical and chemical agent. Please take note also, as we are aware that this virus can be transmitted either to vertical or horizontal transmission, meaning that directly from the breeder to the ill chick, or infection also occur horizontally in the farm. The virus has been grouped into 5 molecular species as A to E, and was further divided into 12 serotypes, namely serotype A or species A, serotype 1, species B, serotype 5, species serotype 4 and 10, species D, serotype 2, 3, 9, and 11, and species E, which is serotype 6, 7, 8A, and 8B, with difference in terms of pathogenicity. When you look at the serotype number, when you look at the European and US, there is some variation with regard of this serotype number. However, you are using the International Committee on Taxonomy of Viruses as a standard reference for serotype number. Because strains with similar serotype show almost identical DNA restriction digestion pattern as compared to strain without cross-reaction in neutralization tests with no common fragment. And the virus also is endemic worldwide among poultry species with different serotype strain involving various geographical region. And it seems that there's some issue or some impact on different serotype with regard of cross-protection. FAdV outbreaks have occurred worldwide. You look in early 90s, it occurred in Middle East, Southeast Asia, and Australia. Then it's distributed all throughout the world. We involve the non-America, South America, we have South Africa, Europe, East Asia, and was first reported in Malaysia in 2005. The case of the disease, which is inclusion body hepatitis outbreak, reported in Malaysia in 2005. It occurred in 34 days old broiler chicken with history of poor growth and relatively high mortality. And you look into the gross lesion, the liver was enlarged with area of synthetical hemorrhages. And histologically, this is a typical lesion of inclusion body hepatitis with the presence of, in this case, basophilic intranuclear inclusion body. Under lateral microscopy of the liver, by negative staining, we can see those of the viral particle, while in the tissue or liver in the nucleus of the hepatocyte, we can see those of the intranucleation body. The virus is, in this case, is susceptible in the SPF hemolytic 8 with thickening of the colon-to-alveolar membrane, necrosis of the liver, and interestingly, there is also hydropericardium. And under histological examination, it present of typical lesion of inclusion body hepatitis with this intranuclear inclusion body in the hepatocyte. The virus strain with the serotype 8B caused 100% mortality in SPF chicks. The sample of this was positive for full adenovirus, and for the field outbreak, we do also conduct PCR for IBDV and CAV, and they are actually negative for those viruses. What is important is that, let me block on the slide there, yeah? We can see that the clinical sign of inclusion body hepatitis, when they are observed with a period of 6 to 14 days, usually it is associated with vertical transmission. While in horizontal transmission, they usually observe clinical sign of IBH in the chicks where it is above what is called 21 days of age. This is another example of consequences of IBH outbreak in the country. The case 1, which occurred in day 7, so possibly this is due to the vertical transmission, we show drop in body weight with pale cones and shrunk, and sudden onset of mortality ranging from 0.5% to 1% per day. And case 2, which is reported in 25 days old broiler chicken, with mortality started at 20 days of age, with 0.2 per day and increasing up to 1%. So this is more likely an example of horizontal transmission. When you look at the gross lesion, at the level of pale, swollen, and yellowish, with presence of critical hemorrhage, both in what I call day 7 and 20 days cases. And histologically, they are present of intranuclear inclusion body in hepatocyte, as well as in the choroidal membrane. Immunoperoxidase confirm that actually those inclusion body is belong to full adenovirus, or what you call it, full adenovirus. Through EM, we also detected the virus in the nucleus of the hepatocyte, and it was further confirmed when you conduct what you call it PCR. So this is some example that what you call it vertical and horizontal transmission could occur as a result of high challenges in the field. The disease also can occur in the layer chickens. And this is an example of a 25 to 27 week old layer chicken with gizzard erosion, gizzard erosion, and presence of black vomiters in the crop and gizzard. Bird have reduced feed consumption and dropped egg production with mortality of 2.0%. Histologically, the presence of intranuclear inclusion body in the liver and gizzard and PCR confirm that actually this infection is due to full adenovirus infection. So what we see here is more of a primary role of full adenovirus causing diseases in chicken. But at the same time also, FAD can be a secondary pathogen. Like in this case, they can be either what we call it primary or secondary pathogen. Where the disease outbreak occur in broiler chicken at age of 32 and 40-day-old broiler chicken. At early stage, when the bird was at 22 to 24 days of age, the 10% chicken had unilateral swollen joint and presence of caseous exudate observed in the joint, but apparently the bird responded to the treatment with antibiotic. However, at the 32-34 days, chicks show difficulty in supporting body weight, and necrosis of the joint show no animal finding. And when you see the bird on necropsy, the very typical area of those of ascites, distended abdomen, presence of those gelatinous material or exudate of fibrin, hemorrhages of the liver, and enlargement of the liver. At the same time, you can see this atrophy of the thymus, histologically as well as grossly. And under histological examination of the affected liver, there is a presence of intranuclear inclusion body. On the right side there actually is a PCR that demonstrates that the liver sample from the liver so positive for full adenovirus and chicken anemia virus, but negative for what we call infectious bursal disease virus. So this is bursal fabricus of the affected chicken, which is look very normal. So this case, indeed, a case of full adenovirus with a multiple infection with chicken anemia virus and CCRD. Whether FAdV as primary or secondary is still a question, but yet this is serotype 8B, which can cause what we call it primary infection. Phylogenetic analysis of most of all the isolate that we have in the country demonstrated that our isolate, whether through hexagon on gene fiber analysis, belong to group E, which is actually serotype 8B. Currently, we only identify what we call it serotype 8B from the isolate we isolated from the country. In terms of prevention and control strategy, this is very important and challenging. What is more important with regard of prevention and control strategy? One is in terms of accurate diagnosis. Accurate diagnosis is so important to ensure that the prevention and control strategy of diseases is effective. Second one is vaccination. We need to have effective vaccination and monitoring program, but not forgetting we need also a good biosecurity and biosafety, because to prevent susceptible chicken from becoming infected, we need a good biosecurity, and to reduce number of susceptible birds where this effective vaccination play the role. So a concept of diagnosis, I mentioned that it based on history, clinical sign, gross inclusion, detection, isolation, identification, and characterization of the virus, either using conventional and molecular techniques. With 12 serotypes and with poor maybe cross protection among the serotype, characterization of any number isolate is important to ensure effective control and prevention strategies against the disease. This is an example, experimental condition with regard to vaccination in broiler chicken using inactivated and live attenuated full adenovirus. Group A, which is only inactivated, Group B actually only attenuated, while Group C is attenuated booster with inactivated, and D is a control where the vaccination is occur as a hatchery vaccination and booster at 14 days of age. And the final species actually is a serotype 8B. When you look there, after hatchery vaccination with the inactivated, the titer increase at 14 days and further increase, slightly increase at day 28. Well, when you look into of the inactivated or the attenuated, there's not much changes there, but probably this could be as a result of the, what we call it, attenuated virus itself. But combination of attenuated and inactivated, when we booster at day 14, we can see there is high concentration of FAdV titer at day 28. It seems that, of course, as normal practice, priming booster of attenuated vaccine with kill vaccine give a better effect in terms of antibody production. And this is an example of the field trial. We look in terms of a vaccination of the breeder chicks at 18 or 16 weeks of age, and serum sample collected for ELISA at 25 to 37 weeks of age. Again, the seed virus is mainly a species E of serotype 8B. You can freely say that when the vaccination was conducted at 18 weeks, there is a high, quite homogeneous antibody titer above 10,000 at 25 to 27 weeks of age. When the vaccination was conducted at 16 weeks, there is some variation, but still very high antibody titer above 6,000 at 25 to 27 weeks of age. The main objective of vaccination of the breeder chicken is to give a good monthly antibody in the progeny. And the second one, of course, it might also necessary to vaccinate the breeder chicken for the protection of the breeder chicken itself against full adenovirus infection. Full adenovirus are often reactivated during point of lay or peak egg production as a result of increased stress. So as a result of that, there's a high potential of transmission of the virus in the progeny or in the day-old chick. High uniform neutralizing antibody can be achieved by priming booster of homogeneous strain to the circulating strain. In this case, what we have is single vaccination, but currently also we are still monitoring in our experiment to give a booster vaccination at a 26 or 28 of age with an objective that we can have high and protective level antibody during the peak production, which is about 34, 35, 37 weeks of age, to ensure that the progeny or the old chick will be protected against the virus. So booster might give a better, protection against the disease. In conclusion, as we're aware that full adenovirus infection is common in all poultry production. And this full adenovirus can be a primary pathogen as well as a secondary agent. It is well established that type E, serotype 8A, which is present, and 8B, where 8B is present in the country, is the main cause of inclusion body hepatitis. Type D, which is serotype 2 and 11, also cause inclusion body hepatitis, and type C, which is serotype 4, cause HHPS. Chicken can be infected with multiple full adenovirus serotype, and it can cause death as well as stunted and poor growth. The virus can be transmitted either through vertical or horizontal transmission or both, and stress and hormonal change can reactivate latent full adenovirus. Particularly, this is important in a broiler breeder or in the breeder chicken. And full adenovirus also can be a secondary agent to immunosuppressive diseases. They are so important, meaning that in control of this disease, a part of focus we given to the virus itself, we might look also in terms of effective vaccination against other immunosuppressive diseases. Unfortunately, limited commercial vaccines are available, and breeder may need multivalent autogenous vaccine. Limited cross protection between serotype, multi-strain vaccine is required and should include prevalence circulating strain. Vaccination of breeder is a major tool to control full adenovirus infection associated diseases in broilers. The issue is that ELISA has limiting meaning as it is only show group specific antibody titer, which is group 1, unless use homologous antigen. Serum neutralization tests detect neutralizing antibody to specific vaccine serotype. Unfortunately, they are not easily available. With that, thank you very much.
Thank you very much, Dato Prof. Hair. That was a wonderful presentation of a very complicated subject, this inclusion body hepatitis. Unfortunately, not many farmers are familiar with the disease still. Now, Dato has shared with us many aspects of the disease, including the taxonomy as well as the epidemiology of the virus, and also presented some of the important clinical signs and postmortem findings on the disease. In particular, I think we'd like to draw the attention to some of the things that Dato has mentioned, and that this particular virus can spread both vertically as well as horizontally. And therefore, in the prevention of the disease, vaccination of the breeder flock is an important step, an important tool in the disease prevention. Dato has also mentioned that good biosecurity and biocontainment are very important, and at the same time also that this disease can occur as a primary pathogen or a secondary pathogen, and that therefore the disease, therefore the biosecurity as well as flock health management to have a good control of the poultry health situation in the farm is very important. And I think another important point to note that it is also a disease that can be brought about by stress as well as hormonal changes in the flock. So I think good biosecurity, reduced stress, good control of poultry good health, these are very vital and important. And I think the vaccination, of course, Dato has touched on both the oil adjuvant as well. There's a water-based, alum-based vaccine available also. And I think we heard from an authority today on this particular subject. I think we will not handle any questions and answers at this stage of the webinar, but we'll reserve questions and answers to the end of the webinar. With that, I will conclude this particular sector now, and I'd like to move on to the next topic, which is also the speaker for the next topic is also Professor Dr. Mohd Hair, and he's going to speak on infectious bursal disease, or IBD for short, and a common name is called Gumboro disease. So again, back to you, Dato.
Thank you, Dr. Yap. Once again, ladies and gentlemen and participants, I would like to share with you on the issue, challenges, and strategy for prevention and control of infectious bursal disease virus infection in chicken. Infectious bursal disease, or IBD, or Gumboro disease, is a virus disease of chicken associated with, first, high mortality, and second one is severe immunosuppression, partly due to the effect of the virus, particularly in the bursal of prebicus, leading to highly susceptibility to secondary infection. IBD outbreak was first reported in 1957 due to the classical infectious bursal disease virus and spread to US and Europe. And in 1986, second strain of IBDV was reported that occurred in Holland, which is actually very virulent infectious bursal disease virus strain, which caused high mortality, and has spread in UK and to most of the country, including Asia, and occurred in Malaysia in 1991. About the same time, in 1980s, there's a new strain of infectious bursal disease virus being reported in USA, and this is known as variant IBDV. And this variant IBDV caused severe bursal atrophy and immunosuppression. Recently, with the analysis in 2017, we have a new strain of a variant IBDV, which is known as variant very virulent IBDV. Indeed, it is an issue and challenges for us to ensure more effective strategy in control of the diseases, again, all the virus strain, but also this new recent strain of variant IBDV. This virus is belong to Barenia virus, which is non-enveloped, with isohexocyclical with diameter of 55 to 65 nanometer. They are double-stranded RNA on content of variant protein of BP1, BP2, BP3, BP4, and BP5. The major capsid protein BP2 is a type-specific antigen and contains the virus neutralizing epitopes. Currently, about 60 to 76% of IBDV isolate are of virulent IBDV. However, when you look carefully, there are significant differences between the African, European, and Asian VVIB strain, suggestive that of independent evolution. The live attenuated vaccine developed against classical IBDV strain failed to respond against the variant IBDV strain and could not provide full protection against very virulent IBDV. The virus itself is a double-stranded RNA virus. The challenge is that these double-stranded RNA viruses are subjected to frequent genetic mutation, the assortment of genome segment, and genomic recombination event that can be potentially increase virulent and alter antigenicity, rendering vaccination efficacy. The major capsid BP2 protein play a vital role in genetic variation by which the virus can escape neutralizing antibody. Highly contagious, the virus is highly contagious and very resistant to chemical and heat activation because they are a non-enveloped virus in which they can persistent survival in the poultry farm up to 54 to what we call it 122 days. Bursal fabricus is a target organ of the virus will lead to risk of irreversible, impermanent damage of the organ leading to immunosuppression. There are two serotypes of IBDV, which is serotype 1 and serotype 2, in which that only serotype 1 is pathogenic in chicken. The classical serotype 1 can cause 50% mortality in SPF chick, where the virulent one can cause 100% in SPF chick, while the variant mortality less than 5%. All the virus strains can cause immunosuppression in the affected bird. Canonical disease has been associated only with serotype 1s against which all commercial vaccines are prepared. Virulent IBDV and classical IBDV strain are common worldwide and cause serious disease. Recently, infectious Bursal virus has been suggested to be classified into 7 serogroups. Previously, we have only what we call it 3 classical in terms of the strain, but now in 2017, it has been suggested to have 7 serogroups based on the marked changes in the amino acid and the BP2 region. The genogroup 4, 5, 6, and 7 have mutations that are likely to contribute to alter antigenicity. Of course, this has become an issue and challenges in terms of control and prevention of those diseases. Study conducted by Jetwood from a 19 sample from 20 countries located in North and South America, Europe, and Asia has classified that infectious Bursal disease genotype into 7 major genogroups. This only paper reported in 2017. The group 1 actually is genetically classical IBDV virus, or classical strain of IBDV. Group 2 is primary antigenic variant virus, which is a variant IBDV. And more importantly, group 3, which is actually very virulent IBD pathotype and very virulent IBD reassortment. It occurs worldwide, but more often outside non-America, Africa, including what I call it, Algeria, Colombia, India, Indonesia, as well as Malaysia and Vietnam. While the group 4, reported in United States, Emirates, and closely related to South America. Group 5 is Mexico, resulting as a recombinant between classical and variant viruses. Group C reported in Kingdom of Saudi Arabia, and Group 7 in Australia and Russia. So when you look at the outbreak of IBDV, was started in 1957, but when you look back about 50 years back, from January to June 2005, 2015, 2010, 2019, the pink there is an incident of the outbreak reported to OIE, while the green there is the country which is free from IBDV. In general, I could summarize that from 2005, 2015, 2010, and 2019, the incident or reported case of IBDV is reducing. I mean, they are less than a year before. The question is that, are the current IBD prevention and control strategy working? The answer can be big yes, but most of the time, no IBD outbreak reported, proper IBD vaccination program, and sometimes also there is no IBDV field challenge. As those cases reported on the OIE also, there's a reduction case of IBD. So big yes. But on the other side, occasionally we can say no, you know? Occasionally, partly because IBD outbreak occur despite of IBD vaccination. And please remember that when there is no IBD outbreak, but outbreak of the diseases and poor performance, probably as a result of immunosuppression due to IBD, still occur, what we call it, from time to time. And I mentioned to you the new challenge again now with the presence of this variant virulent IBDV. Why are the virus so important? I mentioned earlier they can cause sudden death or high mortality. But at the same time also the virus cause immunosuppression, severe immunosuppression as a result of partial or permanent damage of the Bursal of fabricus. When you look back to school, with regard to our immune system or chicken, they have central lymphoid tissue and peripheral lymphoid tissue, where the central lymphoid tissue comprises of Bursal of fabricus and thymus. While the peripheral one, we have the adrenal gland, we have bulk-associated lymphoid tissue, gut-associated lymphoid tissue, spleen, and other lymphoid aggregate in the organ. Infectious disease affecting the immune system of chicken by distracting of the lymphoid organ, particularly the Bursal of fabricus. The target cell of IBDV is a B lymphocyte. In an mature stage and infection, where no factor causes immunosuppression. The B cells are main player in the hormonal immune system, and their depletion leading to immunosuppression, increasing the burst susceptibility to other pathogens and poor or failure of vaccination program other diseases. And I mentioned many of the photos here, the virus also cause infection in other lymphoid organs. You look into the importance of immunosuppression. Today, almost all non-major poultry diseases has been reported in most countries, and you can see also the number is increasing. The success of chicken production depends to a significant degree on the flock health, as well as accurate diagnosis, prevention, and control diseases. Most of the problems related to disease in the chicken industry today are caused by interaction by many factors, where immunosuppression play a key role and is a frequent problem in chicken production. An example, we have an outbreak of ND in year 2000, and this is associated with failure of ND vaccination as well as subclinical IBD infection. Similarly, 10 years later, in 2010, we have another episode of ND outbreak, and as a result also failure of ND vaccination, as well as failure of IBD, as well as ND vaccination. So we can see that during these two outbreak experience, lesson learned, when you of the organ of the chicken affected during the outbreak, you can see how bad is the Bursal of fabricus damage and seem be permanent. And looking at those tissue, we do believe that they can cause permanent damage of the organ and leading to immunosuppression to the chicken. Prevention and control strategies, as mentioned in the previous presentation, the first important thing, again, is accurate diagnosis. I may repeat many times, control and prevention of diseases, accurate diagnosis is so important. And the principle of the accurate diagnosis might involve history, clinical sign, gross and installation, as well as detection, identification, and characterization of the agent either using conventional or molecular technique. So with the new modern molecular biology, in very serious challenges or very serious cases, apart from doing to other conventional, I think molecular technique, particularly characterization of the virus, is needed. Prevention and control also involve to ensure that the interaction between host, environment, and agent is balanced, because disease is adverse interaction between the host, agent, and environment. It means that we need proper biosecurity. Second, we need that cleaning and disinfectant. And we ensure that the host receive very effective vaccination program. Not forgetting, not just to do vaccination, but we need to do also effective monitoring program. I think this sometimes forgotten, to do effective monitoring program. And try to ensure we are having a quality chicks, day-old chicks. And I mentioned earlier, characterization of the virus has become an important now. And with the technique available, I think when we have cases which is more severe, we might do, apart from other lab tests, to characterize the virus where it belongs to, particularly when you have now a new challenge of variant, variant IBDV. Again, to prevent susceptible chicken from becoming infected, not allow the agent to enter the farm, to get contact into the chick, to cause diseases, we need a good biosecurity. But once the virus enters the farm, failure of biosecurity, the virus get into contact with the host, again, failure of biosecurity. This is where actually our vaccination program or strategy is being challenged. If our vaccination is working, the bird is protected. If the vaccination is not working or the program is not right, the chicken will down with the disease. So when considering vaccination, at least there are 5 important factors to consider. One is time of vaccination. Second is the IBD seed virus, our vaccine, the pathotype and the genotype. And then we have the IBDV field strain, type of the type of the vaccine, as well as type of a chicken. I think we have experienced this IBDV since 1991, which is almost 30 years from now. In principle, we control the disease very well, but the challenges and the issue is still there. We introduced quite a number of different types of vaccination. We include of in ovo, using attenuated live, kill, immune complex, and even now we have also what we call it recombinant or vector vaccine. I think the advancement of vaccine and vaccination of infectious bursal disease or Gumboro disease is there. But I mentioned earlier, the issue and challenges is still there as well. In conclusion, Gumboro or infectious bacterial disease is very important diseases in poultry. IBD is a double-stranded RNA virus and is subjected to a frequent genetic mutation, reassortment of genome segment, and genomic recombination event that can potentially increase virulence and alter antigenicity, rendering vaccine less effective. IBD genotypes, we have 7 genotypes there, and about 60 to 70% of IBD isolate are of variable strain or genotype. There are significant difference between the American, European, and Asia of IBDV, suggesting on the independent evolution. The target cell of IBD is a B lymphocyte in Bursal of fabricus, and it might lead to high mortality and risk of irreversible immunosuppression. Cross-protection among classical and variable IBDV is not full, while with a variant IBD is poor. How about the variant IBDV? The whole process for a correct diagnosis should include history, clinical sign, gross and historical lesion, as well as detection, isolation, and identification, as well as characterization of the virus. Prevention and control strategy of Gumboro of IBD. First one, I repeat again, you should have accurate diagnosis. Second, effective management practice. Third, effective biosafety and security practices. And the fourth one, effective vaccine and vaccination strategy. We're lucky now because in terms of R&D, they have developed IBD vaccine based on IBD strain. At the same time, development of new generation IBD vaccine, whether the recombinant or vector vaccine is available. With that, thank you very much.
Thank you very much, Dato Professor Hair Bejo. I think what an awakening for everyone. Dear participants, ladies and gentlemen, we have often heard about HIV in humans, AIDS, the acquired immunodeficiency syndrome in humans. Now today, you hear a little bit about immunosuppression, infectious diseases in poultry. So even in poultry, we have AIDS. But we are very lucky here in a way that we have some adequate preventive and control measures for this particular disease in poultry, or IBD that we call it, infectious bursal disease. Now, Prof. Hair has already given us a very good and comprehensive roundup on the epidemiology of the disease, as well as highlighted some very important points, which I think we should all take note when we are dealing with viruses. And one of the things here is the mutation or genetic reassortment on the genetic genome segments in the virus, creating new variants of the virus. And I think this is something that it's a very important in the field of virology, whether we are dealing with coronavirus, SARS, or we're dealing with influenza virus, another very hot topic, which we'll have another webinar on that, which are also very labile virus and can also mutate quite easily. And even in this particular infectious bursal disease virus, we have such a possibility of this kind of mutation and genetic reassortment, and giving rise to new challenges, causing very virulent strains of the new agent. We have heard from Professor that we have some very effective prevention and control measures on this particular disease, and which we must carry out, because if we don't, the impact is very, very severe, because as it is an immunosuppression disease, it can cause all our other efforts in vaccination, such as farmers will vaccinate their chicken against a very important disease called Newcastle disease, or ND. And if you have IBD around, then your ND vaccinations will not be effective. So I think this is something that's very many farmers must take care of very diligently. And I think we must also remember that besides vaccination, some other very important prevention and control measures which Dato has highlighted, and that is the very accurate diagnostic methodology is required, so that we know what we are dealing with, which strain of virus we are dealing with, and why is our ND vaccination not working, and so on and so forth. Because without proper diagnosis, we will not likely be having good control programs. Dato has also shared with us that the importance of preventive measures also include very good biosecurity, cleaning and sanitation. These are very important. And as well as in the vaccination program, because of the mutation, maybe to have perhaps to look at what they call recombinant combination vaccines, so that combinant vaccines, so that it takes care of more than one strain. I think these are some of the important issues about this disease, infectious bursal disease, our poultry equivalent of HIV, human immunosuppression, immunodeficiency virus in people. So I think with that, we'd like to thank Dr. Professor Mohd Hair Bejo for his two excellent presentations, and we'll probably come back to you on the Q&A sessions later after the second speaker. Now, our second speaker today is Professor Dr. Abdul Rahman Omar. He's currently the Dean of the Faculty of Veterinary Medicine in University Putra, Malaysia. Prof. Rahman has obtained his Doctor of Veterinary Medicine from UPM in 1991, and he has also obtained his PhD in immunology from Cornell University in USA, a very distinguished university in one of the Ivy League universities in the USA, and he's currently with the Faculty of Veterinary Medicine, as I say, as a dean now, but he had been a lecturer before on immunology and infectious diseases. He's also a director of the Institute of Bioscience at UPM and the principal investigator at Laboratory of Vaccine and Biomolecules. Prof. Hair has more than 20 years of teaching and research experience in avian disease and health and production. He's also currently the regional advisor of Global Alliance for Research on Avian Diseases, and contributing all his expertise on various fields in the control of avian diseases. He's also a member of the advisory board of Avian Pathology Journal, and is published in many journals, many articles related to poultry health and diseases. And without much further ado, I'd like now to invite Professor Dr. Abdul Rahman Omar to deliver his speech.
Thank you very much, Dr. Yap, for very kind remarks. Bismillahirrahmanirrahim. Assalamualaikum, and a very good morning to all our distinguished members of this webinar. It is indeed an honor to be with all of you this morning. And okay, let's see. Basically, as what have been stated by Dr. Yap, this webinar is actually focusing on all those various important poultry diseases. And for this morning, I'm going to share with all of you my presentation on infectious bronchitis virus. Okay, a little bit on to focus on the evolution, infection, and immunity. Okay, all right. Okay, these are the outline of my presentation. I'm going to talk about the virus itself and a little bit on the genotyping, especially the classification of the virus. We need to know what are the virus that we have in Malaysia, and also to focus a little bit on the spectrum of IB infection, a little bit on diagnosis, immunity against IB, and then vaccination against IB, and then to summarize the presentation, my presentation this morning on infectious bronchitis virus. Okay. All right. So infectious bronchitis virus is actually classified under the family of coronavirus. And of course, probably now, because of the COVID-19 pandemic with COVID-19, a lot of people now knows a lot about coronavirus. Coronavirus. Currently, coronavirus can be divided into 4 different genus. 4 different genus. And for even for infectious bronchitis virus, they are under the genus of gamma corona. Okay, so this particular slide showing the phylogenetic analysis of the coronavirus, showing the genus as well as the lineages. Okay, so infectious bronchitis virus classified under the genus gamma coronavirus together with the Turkey coronavirus. There's only two family. Okay, this is under the genus gamma, gamma coronaviridae. Okay, and of course, among all these genus of this coronavirus, we have all these alpha, beta, and gamma, delta. The SARS, the SARS-CoV-2, SARS-CoV-1, all those MERS viruses affecting human is actually under beta, and under the alpha we have all those viruses, the feline coronavirus, the virus that causes the deadly disease called feline infectious peritonitis virus. So currently some studies have indicated that the gamma and the delta coronavirus under the gamma delta coronavirus, they derive the gene pool mainly from the avians and pigs, because what happened, pigs also, they have a fair bit of coronavirus, for example, the TGE and everything. Meanwhile, the alpha and beta genus under this coronavirus, they've been speculate they comes mainly from bats. Okay, all right. Okay, if you look into infectious bronchitis virus, this virus exists in a very wide range of antigenically and as well as genetically distinct group of viruses, and this makes control and prevention of this virus can be very challenging. More importantly, chicken is actually the natural host for this virus, and at the same time, beside infectious bronchitis virus, there are many IB-like virus or other avian coronavirus that can be detected in various species of avian, either domestic or wild, such as geese, pigeons, partridge, guinea fowls, and ducks, and so forth. Recently, we have one study, carried at our Faculty of Veterinary Medicine, looking into the detection of avian coronavirus or IB-like virus among all these non-poultry birds, indicating that besides IB, there are many other avian coronavirus that have been not well characterized that can be detected on the various species of birds. Okay, if you look into infectious bronchitis virus, this is actually the RNA virus, a single-stranded, a fairly quite large genome, about 27 kilobases. It has about more than 50 different serotypings, 50 different serotyping. And this is actually the structure or the cartoon of infectious bronchitis virus. These are all those key main antigen protein that express on the virus. One of the most important protein is actually spike protein. Spike protein, they have two, S1 and S2, but the most important is the S1. The S1 protein, they express or they encode for several epitopes or antigenic determinants that play a very important role, especially in terms of immunity. And the S1 also play a very important role in terms of classifying the virus based on antigenicity as well as based on the genotype of the virus. So the S protein is actually one of the most important protein. Probably you heard now the COVID vaccine, the mRNA vaccine that been produced by Pfizer, by the mRNA vaccine, they are actually targeting the S protein also. Okay, right. So if you look into this S protein or this spike protein, why they are so important? Because this protein, this spike protein, it encodes for serotype specific antibody as well as neutralizing antibody that are important to give protection. So what happened at the moment when we have an IB virus in the field, if the virus undergo changes, mutation, and if those changes are actually significant enough, you can have a problem where vaccine that are currently available that have been used will not able to give full protection. However, to determine whether a vaccine really works or not, we really need to determine cross protection studies looking into the use of the vaccine against that particular field strains. Currently, the variation that happened in the S1 gene depends based on the quantity as well as quality of the variation. Some level of cross protection can be obtained. The S1 gene also play a very important role, especially in determining the tropisms of the virus, because now we know some IB virus can actually affect kidneys. It can go to reproductive systems and so forth. So it all associated with the S1 protein. Okay, so one of the important phenomena about the IB virus is actually, I want to introduce the term called what we call as quasi-species. Because it's phenomena of this quasi-species, the virus tend to persist quite significantly, and it will undergo evolution every now and then. But that's why whenever you have infection with the IB or you have a vaccine strain, in the birds, the virus can undergo changes. They can undergo changes where you can see undergo changes either in a form of mutation and recombination. IB virus, being an IB virus, is actually a virus that frequent undergo recombination, where this recombination can happen between a vaccine strain with a field strain, or you can have a recombination between a field strain. And when you have these changes happening, these changes are happening because partly because of the immune pressure that been provided by the vaccination, co-infection with the various strain, and every now and then selection can happen, and this may end up producing a new strain of IBV. And this particular new strain of IBV, if they are significantly different, especially at S1, it can actually complicate our vaccination strategy. Okay, so technically, if you look into IBV classification, can be very challenging, can be very complicated, and can be very difficult to understand. But what I'm trying to do now is basically try to summarize what is actually the best, some of the easier way to understand some of this IBV classification. Generally, they can be classified based on serotypes or genotype, where we have the so-called mass strain. This is what we call as a classical IBV. Some of the strain like M41, H120, and so on and so forth. So these are some of what we call a classical IB strain, or the so-called mass strain. And then we have the so-called non-mass strain, a very huge group of IB viruses, such as the 491, the K2, the D27. Over in the US, we have the Arkansas strain, the Delaware strain, and so forth. So these are all what we call as a non mass strain, and some studies indicate this as a variant strain. Okay, all right. And similarly, we have also another group of we can also have another group of IB that we classify. This is so-called variant strain, and many of these variant strain can be a local variant strain. And depending on the type of these viruses, you can have this so-called local variant strain that are unique for certain countries or region. And then under this so-called variant strain, we have also some very extensively distributed IB virus such as QX strains and so forth. So these are some of the easier ways how we can actually classify IB virus based on mass, non-mass, and so forth. Okay, and the distribution of IB virus, if you look into distribution, pretty much quite distribute based on the geographical region. Over in North America, we have a very unique strain of IB that seems to be affecting various states in United States that we seldom detect in other parts of the world. Likewise, if you look into in South America, also they have some of the strains that have in North America, but at the same time they have this European 793B strains and so forth. Meanwhile, in Europe, you have this various strains of MERS, the European 793B, the 491, the Italy, the QX, and of course the local variants. Same goes in Africa, and in many parts of Asia, we have this mixture of these MERS strains, European 793B, the QX, the L1, and the local variant. Local. So these are basically in Asia. The IB virus is quite heterogeneous. And of course, in Australia, they have a very unique IB strain. They have this so-called variant IB that seems to be detected mainly in Australia. Okay, so what I can conclude from this slide, some of the IB strains are very ubiquitous, meaning that it can detect it with a worldwide distribution. Meanwhile, some of the IB strains are with a pretty much very limited distribution. Okay, probably by now, when you read into literature about this so-called IB classification, you will end up having this very confusing terms called serotypes, genotypes, and prototypes. So what I want to do now here is basically try to give you an idea what does this really mean. As I indicated earlier, you have more than 50 different strains of IB. It can have different antigenic as well as genetic profile. When you do classification, when you look into the antigenic features of this virus, you call it serotypes. So that's why you have IB serotypes. Meanwhile, if you look into the genotype of the virus or genetic of the profile, you call it genotypes. Meanwhile, if you do a vaccine study, meaning that if you do a vaccine challenge studies, you have a particular strain of a virus of IB, and those IB virus strain able to give protection against the field strains, generally we call it a prototypes, meaning that a particular strain of IB will able to give protection against that particular field strain. Because even though we have this so-called very three general ways of classifying this IB, things can get a little bit complicated. Why? Because when you have this classification based on genotype, serotypes, and prototype based classification, they do not always add up in the same way, right? Because they can have different. They will not have similar results and so forth. That makes IBV classification very complicated. Okay, all right. Okay, and in terms of disease spectrum for IB, generally it causes a respiratory disease, affect all age of birds, all strains, and of course it replicates in the trachea and in the lung. Some strains of IB, especially, they can also affect the kidney, cause nephritis. Some strain of IB also can cause enteric problem, affecting the enteritis, and also we have also some strains of IB that actually attack the reproductive tract and cause severe losses in terms of egg productions, in terms of poor egg qualities, and so forth. Another important aspect about IB is actually they are very prone to secondary infection, especially with E. coli, with Mycoplasma, or to some extent with other viruses. When you have this co-infection with this other organism, it can actually increase the severity of IB infection. Okay. Right, diagnosis of IB, currently there are several ways to diagnose IB. I would say that one of the most important, most accurate diagnosis is basically by sequencing. Meaning that if you do sequencing, you do PCR, you detect the S1 gene, and if you do sequence of the S1 gene, you should be able to determine the genotype or the lineage of the virus. If you want to do isolation, very straightforward. You can isolate the virus in embryonic chicken eggs. You inoculate to the SPF, embryonic chicken eggs, where you can notice dwarfisms or mortality of the embryos. Or another way is basically you can inoculate into organ tracheal organ culture. You can see celiostosis, because one of the characteristic features of IB, if you do infect into a tracheal organ culture, you can see actually celiostosis. So one of the important things to diagnose IB is actually to monitor the tracheal health, meaning that if you actually do tracheal culture, you should be able to determine whether your birds are actually well protected against IB or not. Because if you're not able to have good protection against IB, they may develop prone to secondary bacterial infection and everything. So to conclude, in terms of diagnosis, I would say that the best strategy to diagnose IB is actually by PCR, and then followed with a histopathology to determine whether you have problem with IB or no. In case you suspect a variant form of IB, the recommended method is basically we need to isolate the virus using SPF birds. Okay, in terms a little bit on IB in Malaysia. In Malaysia, we first report infectious bronchitis virus in 1967, actually, 1967, and then with the introduction of the use of vaccine, the mass vaccine in early 1980s, and then we start to see increased cases of IB. Where I think one of the well-characterized studies at the time in early 1990s is a strain called MH5365. This is isolated in 1995. This is actually a nephropathogenic strain. And then we have also another strain called V9. So these are some of the very limited studies that have been carried out on the detection of IB in Malaysia. The last couple of years, at least about 5 years, about 7 years back, we also have started to do a lot of surveillance studies to detect IB in Malaysia. What we can actually notice, we have detected a Malaysian variant IB, Malaysian variant IB. This virus is still been detected in the field at the moment. Beside the Malaysian variant IB, we are also detecting a QX-like IB virus that have been circulating in Malaysia. I would say that currently, beside the vaccine strain, the QX-like as well as the Malaysian variant, these are the two strains that have been detected every now and then from the field. Okay, if you look into the phylogenetic analysis, so we know we have this QX-like IB and also this so-called Malaysian variant. So this is what the virus the phylogenetic analysis of the QX and the Malaysian variants. The Malaysian variant that we have at the moment here, it seems to be quite resembling with the MH5369 virus that was detected in 1995. It is good to know that the virus that was detected in 1995 is still circulating in Malaysia. However, you can see over here, there are some variation between the Malaysian variant that we detected now compared to the one that was detected in 1995. So these are the Malaysian variant. And of course, this is the QX. We call it QX-like. Malaysian that have been detected also in Malaysia. They've been located similarly to the QX strain that have been that are detected in China, and all these other groups. These are basically other strains of IB that have been detected. This phylogenetic tree based on bootstrapping, 1,000 bootstrapping. You can see actual classification of the virus based on the different geographical regions and so forth. Okay, all right. Okay, another important aspect in terms of classification of IB in 2016, Valastro tried to harmonize how we can actually classify IB, and where they propose identification of IB based on 6 genotypes, 32 lineages and few lineages based on recombination. So under that particular, this so-called G1 genotype, they have 27 lineages, and then the remaining genotypes actually has 1 lineages. However, since 2016, even though Walsro have put up this classification, the number of new IB lineages or new strains of IB have been detected, and we end up having several different strains of IB that are unclassified. Okay, so we also did similar analysis based on our Malaysian variant strain and QX strain that what we have. At the moment, this is based on the Walsro classification of IB. We like to share over here. The Malaysian QX strains actually been grouped in G119. This is in this particular group, together with all those QX strains, this LX4 strains. These are all QX-like virus that been detected. So the Malaysian QX strains are actually in this particular group. Meanwhile, the local variant strain seems to be quite unique under the so-called Walsro strain classification. It's still not being categorized in any other groups. The G1, this is the mass strain. Meanwhile, this G113, this is actually all those 793B and so forth. So this is basically, again, to indicate that IB is actually a very virus that undergo significant mutation, variation, and even though a new classification of IB here unable to categorize or unable to group all those new strains of IB that we have detected recently. Okay, so we also did some studies in terms of this QX Malaysian strains as well as this Malaysian variant strain. What we can tell, the Malaysian QX strains are more pathogenic than the Malaysian variant strain. However, both strains actually affecting the respiratory and also the kidney. So we also have did a complete genome of the Malaysian variant strain. What we can indicate, it seems is a recombinant strain, probably with the parental strains of this 491, and of course the QX strain, the Malaysian QX strain is also a recombinant strain. They have a very similar features to the QX strain that was first detected in China. So we also did some studies looking into whether vaccination with our mass or the so-called non-mass or variant strain, can it give any protection against this Malaysian variant of QX? So what we did single vaccination, either with mass or non-mass strains, and then after 2 weeks, after 2 weeks of vaccination, we challenged them with the Malaysian variant strain and a QX strain, and we measure protection based on Serotypes. We are just looking into whether the vaccination can actually give protection against Serotypes as no. What we notice, we can detect a partial protection against this single vaccination. So this is actually I like to share in this trial. The non-vaccinated birds have a very good celiac activity approaching 100%. The non-vaccinated challenged bird. Meanwhile, the non-vaccinated challenged bird, if you challenge them, will have a very low celiac activity between 14 to 16%. This is basically once you challenge them with the Malaysian QX or with the Malaysian variant. Once you start vaccination, single vaccination with all these different strains of either mass or non-mass, you can see a protection between 50 to about 59% of protection in terms of Serotypes, indicating that if you do a single vaccination with mass or single vaccination with non-mass, you may not able to get a good protection against the mass, against the Malaysian QX or with the Malaysian variant. Probably we need to look into a combination of a IB strain in order to give good protection against these two strains of field Malaysian IB strains. Okay, so we also did. So we tried to answer question why we are seeing this kind of efficacy study. So when we did virus neutralization assay, so this is where we have antibodies that been raised against all these various mass and non-mass strains, and we do a cross virus neutralization assay. We can notice some variation in terms of the antigenicity, indicating why we are seeing this partial protections against this so-called Malaysian variants, as well as against this Malaysian QX-like virus. So these studies actually to look into, we want to map what are actually the epitopes so that we can actually know what are the best vaccine that actually we can actually recommend against the so-called Malaysian variants and Malaysian QX strains that we have at the moment. Okay, so these are basically some of the commercial vaccine that have been registered. Currently, I got this one from the Department of Service. Majority of the vaccine is actually mass and actually non-mass. About 5.8% is actually a combination of mass actually not variant strains. So these are basically vaccine that currently we have, and it is very important. These strains of vaccine that we have, we need to make sure that they're able to give protection against the variant strain as well as against the QX variant strain that we have in Malaysia. Okay, so in terms of immunity, as I indicated earlier, the S1, they encode for epitopes or antigen determinants are very important. They're able to induce neutralizing antibodies and protective immunity. And of course, different strains of virus may have different epitope and they may not be able to give good protection. Okay, that's why it's very important for us to choose the right vaccine strain so that it can give protection against the challenge virus. In terms of immunity, it seems that probably local immunity, especially secretory IgA, play a very important role, especially in protecting the respiratory tract, especially when you talk about the protecting your trachea, so that you have a good tracheal health. So you need this very strong secretory IgA. Systemic antibody, especially IgG, probably play a very important role if you want to give protection against the kidney and against the reproductive tracts. Knowing IB is a persistent virus. If you want to have viral clearance, you need a cell-mediated immunity. And of course, to have cell-mediated immunity, you have to use a live attenuated vaccine where you can induce a T-cell response, either a CD4 positive and CD8 positive T cells. And these cells are important in terms of inducing viral clearance. Okay. In terms of vaccination, as I indicated earlier, vaccination, the S1 play a very important role. And currently, we have a live attenuated vaccine and inactive vaccine available. The kill vaccine is able to induce long-term immunity. But then remember, if you use kill vaccine, it may not able to induce good mucosal immunity, as the secretory IgA or the CMI response. So that's why in broilers, we have this so-called vaccination with a live vaccine. And of course, in long-lived birds, a combination of live, most of the time we start with a live, and we follow up with a kill vaccine being used. And by doing this combination, we should be able to get a good protection against this so-called long-life birds. Okay, in terms of vaccination, again, as I said, whenever you have a new strains of IB, variant strain, this may complicate our vaccination. And that's why it's very important whenever we have a new strain of IB, we need to make sure that wherever the vaccine that we're using able to give good protection. At the moment, there's no single vaccine that able to give full protection against different strains of IB. So that's why we have to make sure we have to choose the right type of vaccine antigen. If we can actually find a protectotype strain, meaning that you have a certain strain of a vaccine that able to give better protection than the particular strain, that's basically one of the way to go. Another important aspect, probably combination of more than 2 different strains of IB will definitely give a maximum cross protection. And of course, the best strategy is actually you need to do a cross protection of the vaccine strain with the field strain in order to determine the best strategies in terms of controlling the birds. So in summary, so this is my last slide. For me to summarize about IB, as I indicate, IB is a virus that are highly mutatable. It can undergo recombination. And some strains are very ubiquitous, and meanwhile, some strains have a very limited distribution. In terms of disease spectrum, some of these virus, they can cause subclinical infection, can be either mild to moderate, and some actually can cause persistent infection, and more importantly, can end up having a secondary infection. In terms of management, if you have a very poor management in terms of farm, husbandry, the disease can spread rapidly, can become more severe, especially if we have with secondary infection. In terms of control and prevention, the diagnosis can be a little bit complicated, especially if you're dealing with subclinical and mixed infection. vaccination is a very important component. You need to make sure you choose the right vaccine strains and the right vaccination program in order to get full protections against IB. Okay, right. Thank you very much. For listening to my presentation, so I'd like to acknowledge. So these are all the postgraduate students that have been working in my lab in terms of this particular project, and with Professor Dato Mohd Hair helping in some of the diagnosis in terms of some of the studies on the challenge studies. With that, thank you very much. Thank you.
Thank you very much, Professor Dr. Abdul Rahman, for a very detailed and very good comprehensive speech on the infectious bronchitis virus. We were talking just now about Prof. Hair, Dr. Prof. Hair's presentation on IBD or Gumboro disease, and saying that, wow, in humans we have AIDS, and in poultry we have Gumboro disease. Now, in humans we have a pandemic COVID-19 disease. Hey, in poultry we also have a coronavirus that causes problem, and it's called infectious bronchitis virus. It's also a coronavirus. And so you see the importance of veterinary public health, whereby we are spreading interlink between the veterinary science and the human health. And I think this is a spectrum that the One Health becomes very important in our modern day living. And One Health will make us understand how we have a lot of similarities between infections in animals and birds as in humans. And I think what Prof. Rahman and Prof. Hair today had shared with us are some important reminders on this issue. Now, I think Prof. Rahman has given us a very good comprehensive review on the epidemiology of the infectious bronchitis virus, and again reminded us that the virus has also, just like in the coronavirus in humans, or causing SARS in humans, it has mutated. Those days we have SARS, then we have MERS, and today we have COVID-19. Infectious bronchitis virus in poultry also have many various strains, and also as a result of mutation and recombination, genome recombinations. So I think this is something that we have to keep in mind when we are doing our prevention and control measures. Prof. Rahman has reminded that it is very important that we know what we are dealing with. In other words, we must do a diagnosis, a test, and find out what is the virus strain that we are looking at. Otherwise, your vaccination against IBV will not be very effective. And I think this is something that the farmers and the vets in the industry, flock health management teams will have to pay attention to. That is to cooperate between what you are working with and the laboratories. Of course, go to accredited laboratories. And of course, we also are reminded that both IB, infectious bronchitis, are often coexisting with other diseases, and it can cause a spectrum of clinical aspects, ranging from respiratory to also reproductive functions, as well as other epithelial sites or disease affecting other epithelial sites in the avian anatomy. And I think this is where also it can co-infection with the mycoplasma infections. So you see, the disease by itself is one thing, but the co-infections are very important. This is a very common phenomenon nowadays in our intensified way of poultry rearing in our industries. And I think flock health management must again make sure there will be a proper good biosecurity, cleaning and sanitation, and knowing exactly what you are facing and dealing with. So you have a very close relationship with the universities, with the laboratories, and with the Department of Veterinary Services. And I think, and the first question, want to ask about this from Law. Hi, thanks for a wonderful presentation. I want to ask which serotype can cause gizzard erosion in broiler? It didn't say who he wants this question to be addressed to. Prof.
In our experience, we have serotype 8B, you know, that cause those of the erosion in the broilers.
Okay, so this is you're talking about IBH, is it?
Yeah.
Yeah. Okay. I hope that answers your question, Mr. Law. Anything to add, Prof. Rahman?
No, basically, as what Prof mentioned, we have these different adeno. They have these different tissue tropisms. Like I think myself, Prof also, we are trying to look for other adenovirus. Apparently, in Malaysia seems to be the serotype 8 seems to be the common strains that we are detecting. So we don't have good experience. We have not detected the serotype. Seems to be less serotype 4 or serotype 2. So we have not detected. I don't know, Prof, you have any comment also? We also have not able to see any good cases. But every now and then we see this gizzard erosion, but it's either negative for adeno or end up something else, but definitely not the other two serotypes and so forth. Prof, you probably want to comment.
Yes. Unfortunately, currently in the country, the challenge virus we have is only serotype AB. Of course, we are try hard for other cases, try to look for other possible serotypes.
Okay, very good, Prof, and to the two distinguished professors. I think my next question here is from Encik Faisal Ghazali. Is addressed to Professor Dato Hair. What are the current status of prevalence of IBH in Malaysia? Or is there current outbreak anywhere in the region?
When look in terms of prevalence, of course, currently we didn't do a very good study on prevalence. But when look carefully, I think the first case of IBH occurred in 2005. Then we have quite a number of increasing cases, sometime in 2008. Follow up, I think the most recent one, we have about 2015, 2018, 2018, and 2017, 18, 19. But it seems that the cases seem to be reducing compared with earlier one. Yeah? So if we do have also probably for this year, we have only a handful of cases. I mean, it's not that heavy as what we do have in the earlier years, particularly maybe before years of the highest one, probably that one is on year 2015 and 16. I mentioned earlier, we didn't do a study of prevalence, but looking at the cases that we do have, against, I mentioned earlier that it seems that the cases, particularly in the broiler chicken, is reducing. Thank you.
Thank you, Dato. Now the next question comes from, I think this is Dato Vincent Ng. Why is the titer of vaccinated chicken of attenuated strain at 28D lower than inactivated vaccine? Okay. Do you think that vaccination of inactivated vaccine is enough for protection instead of the combination of the two vaccines? Okay. I think Dato would like to comment.
Thank you very much, Dato Vincent. That one is under experimental condition where we use broiler chicken with monoclonal antibody. Although the monoclonal antibody is not very high, it's about 1,000. Again, I mentioned to you, this is only the experimental condition. Currently, with regard of R&D on the vaccine development, I could say that we are quite successful using inactivated full adenovirus either with well adjuvant or with LM adjuvant. But our R&D on the attenuated viruses using a different isolate, as well as using different cell line and embryonic chicken eggs, it seems that the result is not that very consistent. So I mean that consistent, sometimes they're doing well, sometimes they don't. And what I can show you on those slides, in those presentation, experimental condition in a broiler chicken, the result saying that to show that the attenuated virus that we use is not able to induce antibody as good as the kill one, probably because of the isolate itself, the attenuation itself. Nothing to do with something about this due to the attenuation cell. So looking at that, of course, we can see that using a kill or inactivated is better with regard to those results. But yet it's still controversial against when you want to use live attenuated in broiler chickens, or even also in broilers, because this virus is have a vertical transmission. So I still do believe that the best way to control IBH is through vaccination of the breeder flocks with proper vaccination program in the breeder flock, so as the day-old chick or the progeny will get protected. Until unless it's needed, then we have to think of what we call it a vaccination in broilers. Thank you.
Thank you very much, Dato. Prof. Rahman, anything to add on this subject?
No. Okay. I don't have any additional input. No.
Fine. Now we're going on the next question from Irfan Nasri. There are two questions here. Again, addressed to Dato Professor Hair. It says, may I have opinion from Prof, how do we follow up the antibody titer after the chicken get maximum antibody protection at day 28 of age?
You mean the follow-ups after, I think the question not very clear, but they're asking about after 28 days that the antibody titer is higher. With that antibody titer, I think it's about quite high, and we do believe that will give a protection for the bird throughout the rearing period, which is, I think in broilers, whether you keep it between 35 or even 42 days, you know. With the antibody, the bird will be protected.
Yeah. The second question, Dato, is it advisable for us to give vaccination to chicks as early as day old, especially when the chicken infected with the fowl adenovirus serotype 8B?
Yeah, the current practice of vaccination, I think people are now, many will move to the hatchery vaccination. I mean, from our few trials with regard of this hatchery vaccination, it worked well. And even from those examples I give to you, vaccination in the broiler chicken at day old or hatchery vaccination, the antibody titer outcome from those vaccination seem to be promising, high, and appear to be protective. So yes, I think I will move now, I think, towards more of hatchery vaccination. Thank you.
Thank you, Dato. The next question comes from Lim Kamin. It's a question fairly similar to the first question, actually. Going around the fields nowadays, I notice a lot of proventriculitis issue with gizzard erosion and liver discoloration. May I know adenovirus will cause the above findings, as the samples I sent to lab for PCR tests all came back with negative result.
Okay. So please remember that back to, I think, principle, we call differential diagnosis, isn't it? I think those editions have quite a number of differential diagnoses. And of course, one of them is fowl adenovirus, particularly serotype 8B. But other potential agents and disease also has been considered. because even adenovirus also, we don't have, we call it pathognomic lesion. Those lesions just suggestive quite number of different diagnoses in addition to full adenovirus infection. So we need to consider to come out or look for diagnosis for other potential agent as well. Thank you.
Thank you, Dato. The next question doesn't have a name there, but it is again directed to Prof. Hair. It says a couple of quick questions. It says, is it advisable to vaccinate broiler against fowl adenovirus if the breeder is not routinely vaccinated against the disease? Secondly, question is FAdV fowl adenovirus common in broiler chickens in Malaysia?
The first question I answered before, if we have no choice, we can plan and do a vaccination strategy in the broilers. But the most important to me is still to consider a proper strategies in breeder flocks. With regard of the, what I call it, common, as mentioned earlier in terms of prevalence in the first question just now, yes, adenovirus now is become common in the country in terms of cases and so on, although it seems that the prevalence or the incidence is bit reducing compared with maybe 2, 3, or 4 years back. Thank you.
Another question on FAD virus. The antibody for the breeders and progeny, do they need to be homologous to the challenge strain? Talking about serotype 8B.
Yeah. There's an issue or maybe controversial with regard of ability to provide cross protection among the what I call it FAdV serotypes. But generally, people agree that they are not, different serotypes are not able to provide good protection against the others. Meaning that, to answer that, yes, we need a homogeneous, what I call it, antibody. Because generally, people believe that this full adenovirus of different serotype will not able to give protection amongst the serotypes. This is not good cross protection. Thank you.
Next question comes from Ali Mansour. I think the answer you have covered well already, but let me just read the question anyway. My question is, he says, do we have a vaccine that will provide cross protection against fowl adenovirus?
Currently not yet, but maybe we can have later on multiple bivalent or trivalent vaccine. Okay.
Okay, this is from Dr. Mulao. I'm not so sure. It says, I think Prof. Hair, Dato Prof. Hair, the FAD virus equal to EDS76 adenovirus. Are they cross protection?
If you look in terms of the group just now, the full adenovirus is group 1, while 8-dog syndrome is group 3, you know. So to answer that, they are not likely to give cross protection. Even among the group 1 of the isolate also, they don't give a group cross protection. And this one, EDS is group 3, while IBH is a group 1. So the answer, I think most likely they don't give any cross protection.
Okay. Then let's go on to ask about the commercial broiler vaccines, whether the cost, but I think I would refrain the question because I don't want to go into, in this particular webinar, talking about which company produced what vaccine and at what cost, yeah. So, of course, the FAD virus vaccines are available in the market, and but he says that you recommend vaccination in breeder because it provides maternal antibody do I need to vaccinate against egg drop syndrome 76? I think you've answered that question already.
Yeah.
Limpic share. How to control the vertical transmission in the infected breeder flock. He experienced outbreak of the disease secondary to stress factors. It's present as a latent form, and this stress factor trigger outbreak of IBH. What is your comments, Prof?
I think that I mentioned in my presentation just now, this involved with, one is the vaccination strategy. Usually when we think about in terms of vertical transmission, usually commonly occur in the flock of broiler chicken that is from a breeder age about maybe 34, 35, 36, 37 of age, where it is a time of peak production. And during this time of peak production, there is shedding, chances of high shedding of the full adenovirus if the bird is infected, because full adenovirus can cause latent infection. So as a result of that, during this period, there will be high risk of vertical transmission as a result of stress factor, and people believe also the hormonal factor. So the best thing to do, perhaps, is to, in our vaccination strategy, I think in our trial, what we do, a vaccination strategy, whether you conducted a single vaccination, usually at 18 weeks of age, in which they can give a quite high protective level of antibody during the 26 or 28 of age. And we do believe with that about 10,000 might can retain until during the peak production. But if we are still not comfortable, perhaps we need to do a booster, because it's always believed that booster will give a better antibody titer. So with that regard, we need to give 18 weeks of vaccination of a kill vaccine. Then booster sometime at the 26 to 28 to ensure that the antibody titer will be at maximum or optimum during the peak production. So by doing so, the bird, what you call it, those day-old chick from breeder flock of between 34 to 37 or 38 weeks are fully protected from vertical transmission if the breeder was infected with full adenovirus. If they are not infected, so no problem. Thank you, Yap.
Okay. This next question I'd like to direct to Prof. Rahman. There's a question from Dr. Vincent Ng. I hope you don't mind to comment on the mRNA vaccine for COVID-19, which may not be related to chicken. According to report, the vaccine is only 95% effective, and it cannot neutralize the COVID virus, so leaving the vaccinated infected as carriers. At the same time, the natural infection leave about 98% immunity after natural infection, which is even better immunity. So why vaccinate? Is it safe to use the vaccine? Sorry, Prof. I bring this question up because COVID-19 is such a hot topic today. And I guess that's why Dr. Vincent is bringing this up here. But I think it'd be interesting. What are your comments?
Thank you. I think we know that I think for COVID-19, I think the risk group, I mean, if you are classified under the so-called risk group, people under the risk group, I think vaccination is something I think will be a little bit recommended. That's basically what's the whole entire idea. And of course, and we also do not know whether at the moment, whether are we going to see a very good immunity if you get natural exposure, because now we can see a lot of probably some of us, if we came down with infection or we get develop natural immunity and everything. So that's it. Against, I would say that it's a bit too early for us to make speculation. But I would say that vaccine will be made available, and beside the mRNA vaccine, we have the inactivated vaccine. And of course, the mRNA vaccine is too early to say whether it's going to be protective or no, because I think they just did a very simple first phase clinical trial. First clinical trial is only about 20, 30 people only. So very too early for us to say. And of course, besides the mRNA vaccine, we have the inactive vaccine, and that is also available. And I would say that my point of view, if let's say, for example, in individual, that if you are actually falls under the group of high risk factors because of the age, because of other complications, everything, I think vaccination is probably something of important. And of course, for healthy individual, probably we can actually see how it's going to go. But nevertheless, I think you all see how it, depending on the scenario. I myself also, I a bit worried to travel overseas without vaccination. I don't know what's going to happen. So we'll see how it goes. So, but I'm more comfortable with the inactivated vaccine and not the mRNA vaccine. My comment. Thank you.
Thank you, Prof. Rahman. Another question for you. This again from Lau Muleong. This is thank you for a good update on IB. As a producer, I'm thinking this is more of a statement than a question. I'm taking a commercial view. We are giving many types of vaccines to breeders and broilers, but we're still not getting optimum performance and profit. Meaning, to have successful result involve many factors, such as feed, human resource management, equipment, genetic, and so on. High time UPM should set up poultry center, cheapest protein source, where it involved expert from equipment, feed, diseases, microbiology. What I think preventive medicine, not only by vaccinating approach, what do you think?
Okay, all right, good comment, suggestion, and of course from UPM, we especially we have a fairly good setup for, especially in terms of diagnosis of diseases, and we also have some looking into development of vaccine. I think that seems to be doing quite well, and of course, nevertheless, we can really expand those what are we doing. Definitely with proper collaboration, probably we can have good setups, good with all these various stakeholders. I think probably we can do more. And secondly, of course, beside the disease part, we have all those production parts in terms of how to have good supplies of feed or probiotics or supplements. Again, I think experts around in the UPM, from the vet faculty, from the Department of Immunosuppression. I think this is something that definitely can be explored together. And of course, if we also be able to get good supports and collaboration with all the various stakeholders, with the industry, I think I'm always game for this kind of approach.
Dato, you want to make a comment on this suggestion from?
I do agree of suggestion. Any collaboration is most welcome. Probably what best is even to start with it, we can sit down and discuss, look for common and look for the opportunity. Because we have all the experts in the university, also the expert in the industry, and even many of them also are very, experienced, particularly the stakeholders. I think we should have a platform and probably could start talking about the ideas. Thank you.
Yes, I like to finish my job here as a moderator for this morning, and I'd like to hand the chair back to you, Puan Nadia.
Thank you, Dr. Yap, and all panelists for the sharing on this important topic in poultry industry. Ladies and gentlemen, we have come to the end of our session today. We, Agro-Food Productivity Nexus AFPN, in collaboration with Federation of Livestock Farmers Association of Malaysia, FLFAM, hope that it has been educational for everyone, and the information gained from today's session would benefit you all. It has been a great day and a wonderful afternoon with all of you. Again, thank you and goodbye.